Jingle bell, single bell, jingle all the way. Oh, jingle bell, jingle all the way. सब लोग सो गए क्या आज इतनी जल्दी? 10 ही तो हुआ है. I think everyone slept. Hi, Parth. भाई अलग ही दुख लगता है जब मुस्लिम लोग ऐसे करते हैं तो. हाँ आदित्य बोलो ना. कोई और रिलीजन का होता भी इतना अफेक्ट नहीं करता. कॉफी स्ट्रॉन्ग नहीं था बिकॉज मुझे इतनी ज़्यादा इस्लाम में फेथ है ना, तो मैं नहीं चाहती कि जिस रिलीजन को मैं इतना ज़्यादा मानती हूँ वही रिलीजन का इंसान आके ऐसा कमेंट करे. एनीवेज छोड़ो, वो फॉलो भी नहीं करता होगा इस्लाम को ठीक से. ओह ओके, थैंक यू आदित्य. मैं आदित्य को ना मॉडरेटर बना देती हूँ ताकि कोई मॉडरेटर ना हो ना, तो आदित्य कैन प्रोटेक्ट मी एज मॉडरेटर. Here you go, Aditya. What is your total study hours a day? I don't have any fixed number. Hi नोनू, तुम लेट आई. मैं किसी से अभी अभी ऊपर क्या सुन के बैठी हूँ यहाँ पर देखो. हाय नीति. आई डोंट नो, कोई रैंडम स्ट्रेंजर था शायद. एक मिनट. हाँ मैं ना सब्सक्राइबर मोड पे कर देती हूँ ताकि कोई गंदा ना लिख सके. 20 मिनट. हाँ अब कोई नहीं लिख सकता. Report harassment or bullying. मॉडरेटर ना टाइम आउट दे सकता है। लाइक अगर कोई एक इंसान आया और वो अननेसेसरी या फिर अब्यूजिव वर्ड यूज़ कर रहा है कमेंट सेक्शन में, तो तुम उसको या तो चैनल से कम्प्लीटली बैन कर सकते हो या फिर उसको सर्टेन टाइम से ना किक आउट कर दे सकते हो। लाइक 30 मिनट तक वो इंसान टेक्स्ट नहीं कर पाएगा या फिर 24 आवर तक चैट सेक्शन में पार्टिसिपेट नहीं कर पाएगा। वैसे यूजुअली होते हैं, कोई ना कोई मॉडरेटर रहता है इधर, लेकिन अगर नहीं रहा ना तभी तुम करना। ओके ओके पार्थ, लेट्स स्टडी। मॉर्निंग गुडिंग। 30 मिनट। ओके 30 मिनट से ही स्टार्ट करते हैं पहले का। गुड लक निधि। आदर्श तुम भी हम लोगों के साथ पढ़ रहे हो ना? आई थिंक आदित्य इज ऑल्सो स्टडिंग विद अस अलॉन्ग विद पार्थ। वी ऑल आर स्टडिंग फॉर नीट, गिरीश। ओके गुड लक एवरीवन। 30 मिनट के बाद मिलते हैं। मैं क्यों इतना वियर्ड हरकत कर देती हूँ, कोई बात नहीं, अभी तो थोड़ा एनर्जी है ना। थैंक यू आदर्श। थर्टी मिनट के बाद 5 मिनट का ब्रेक लेंगे। ओके तब तक सब लोग पढ़ाई करो अच्छे से। गुड लक एवरीवन। Gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Hereditary disease. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy to correct hereditary disease. Gene therapy is a collection of method that allows correction gene defect, correction of gene defect, astaghfirullah, correction of gene defect that has been diagnosed in a child embryo. Okay, if a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allows correction of gene defect. Gene therapy is a collection of method that allows correction of gene defect that has been diagnosed in a child embryo. Collection of method that allows correction of a gene defect. Correction of a gene defect that has been diagnosed in a child or embryo. Correction of gene defect that has been diagnosed. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. Diagnosed in child or embryo. Correction of defected gene. Gene therapy that has been diagnosed in a child embryo. Here genes are inserted into a person's cell and tissue to treat a disease. Genes are inserted into a person's cell. Genes are inserted into a person's cell tissues to treat a disease. Correction of genetic defects involves delivery of normal gene into the individual or embryo to take over the function of and compensate for non-functional gene. Correction of a genetic defect. Correction of a genetic defect. Correction of a genetic defect involves delivery of a normal gene into individual or embryo to take over the function of and compensate for the non-functional gene. Delivery of normal gene. Delivery of normal gene into the individual or embryo to take over the function and compensate for the non-functional gene. The first clinical gene therapy was with ADA deficiency, adenosine deaminase deficiency, adenosine deaminase deficiency, ADA deficiency, adenosine deaminase deficiency, adenosine deaminase ADA deficiency, adenosine deaminase deficiency. This enzyme is crucial for immune system to function. Immune system. This enzyme adenosine deaminase deficiency, deaminase deficiency, adenosine deaminase deficiency enzyme. Enzyme adenosine deaminase is essential for immune system to function. The disorder is caused due to the deletion of a this disorder. The disorder is caused due to the deletion of the gene for adenosine deaminase. The disorder is caused due to the deletion of gene for adenosine deaminase. Adenosine astaghfirullah. The disorder is caused due to the deletion of gene for adenosine for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplant. In some children, ADA deficiency can be cured by bone marrow transplantation. In other, it can be treated by enzyme replacement therapy. Either by bone marrow transplantation, cured by bone marrow transplantation, number 1, or by enzyme replacement therapy. Enzyme replacement therapy or bone marrow transplantation. 4-year-old girl, adenosine deaminase deficiency. Adenosine deaminase, okay, enzyme, okay, adenosine deaminase, ADA deficiency, immune system. The disorder is caused due to the deletion of the gene. Deletion of gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In other, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. Bone marrow transplantation. In other, it can be treated by enzyme replacement therapy, in which functional ADA, adenosine deaminase, is given to the patient by injection. Functional ADA is given to the patient by injection. In some children, ADA deficiency can be cured by bone marrow transplantation. In other, it can be treated by enzyme replacement therapy. Bone marrow transplantation, and either it can be treated by enzyme replacement therapy, in which functional adenosine deaminase, is given to patient by injection. Astaghfirullah, why am I thinking about it? In which functional ADA is given to the patient by injection. enzyme replacement therapy, in which functional EDA is given to the patient by injection. Okay. But the problem with both of these approaches that they are not completely curable. The problem with both of this approach, bone marrow transplantation and enzyme replacement therapy. Therapy, but the problem with both of these approaches that they are not completely curative. As a first step toward the gene therapy, as a first step towards gene therapy, first step towards gene therapy, lymphocytes from the blood, lymphocytes, WBC, right? Lymphocytes from the blood of the patients are grown in culture outside the body. As a first step towards gene therapy, first step of gene therapy. First step of gene therapy, lymphocytes from the blood. Lymphocytes from the blood of the patients are grown in culture outside the body. Outside the body. A functional ADA, adenosine deaminase, or a functional adenosine deaminase, cDNA, using a retroviral vector is introduced into this lymphocyte. A functional EDA, adenosine deaminase, cDNA, using a retroviral vector. A functional EDA, cDNA, using a retroviral vector is then introduced into this lymphocyte. Using a retroviral vector is then introduced into this lymphocytes. EDA, functional EDA, cDNA, adenosine deaminase, cDNA, using a retroviral vector is then introduced into this lymphocytes, which are subsequently returned to the patient. Okay. A functional EDA, cDNA, using a retroviral vector is then introduced into the lymphocyte, which are subsequently returned to the patient. However, as the cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocyte. Okay. However, as the cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolate from marrow cell producing ADA is introduced into the cell at early embryonic stage, it could be a permanent cure. Gene isolate from marrow cell producing ADA. Gene isolate from marrow cell producing ADA is introduced into the cell at early embryonic stage. It could be permanent cure. Early embryonic stage, marrow cell isolate from marrow cell. Gene isolate from marrow cell producing ADA. Marrow cell producing ADA is introduced into the cell at early embryonic stage. It could be permanent cure. Gene therapy. Gene therapy. If a person is born with a hereditary disease, gene therapy is an attempt to cure it. Gene therapy is a collection of method that allows correction of a gene defect that has been diagnosed in a child embryo. Gene therapy is a collection of method that allows correction of gene defect that has been diagnosed in a child or method that allows correction of gene defect. Correction of gene defect that has been diagnosed in child or embryo. Correction of gene defect. Correction of defected gene. Excuse me. Diagnosed in child embryo. Diagnosed in a child embryo. Genes are inserted into a person's cell and tissue to treat a disease. Inserted into a person's cells and tissues to treat a disease. Correction of genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the functional gene. Correction of genetic defect. Correction of a genetic defect involves delivery of normal gene into the individual or embryo to take over the function of and compensate the non-functional gene. Genetic defect involves delivery of normal gene into the cell, individual or embryo to take over the function of and compensate for the non-functional gene. The first clinical gene therapy. First clinical gene therapy was given in the year 1990 to a 4-year-old girl suffering from ADA deficiency, that is adenosine adenosine deaminase deficiency. First clinical therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to deletion of the gene for adenosine deaminase. The disorder is caused due to the deletion of gene for adenosine deaminase. Disorder is caused due to the deletion of gene for adenosine. Deletion on this. The disorder is caused due to the deletion of the gene for adenosine deaminase. Deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In other, it can be treated by enzyme replacement therapy. The disorder is caused due to deletion of the gene for adenosine deaminase. The disorder is caused due to deletion of the gene for adenosine deaminase. Deletion of the gene for adenosine deaminase. Delete when there's a deletion of the gene. In some child, ADA deficiency can be cured by bone marrow transplantation and enzyme therapy. In other, it can be treated by enzyme replacement therapy. Enzyme replacement therapy, in which functional ADA, functional adenosine deaminase, is given to the patient by injection. Enzyme replacement therapy using injection. Functional enzyme is given to the patient through injection. Enzyme replacement therapy. They are not completely curable. Both these do not help in completely curing the disease. At first step towards the gene therapy, lymphocytes from the blood of the patient. Lymphocyte from the blood of the patient. WBC from the blood of the patient are grown in a culture outside the body. Are grown in a culture outside the body. Functional ADA. Adenosine deaminase, cDNA, or functional ADA, cDNA, using a retrovirus. Functional ADA, cDNA, using a retroviral vector, is then introduced into the lymphocyte, which are subsequently returned to the patient. However, as the cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. Periodic infusion of genetically engineered lymphocyte. However, if the gene isolate from marrow cells producing ADA is not introduced into cell at early embryonic stage, it could be A. However, if the gene isolate from marrow cell producing ADA is introduced into cell, introduced into cell at early embryonic stage. It can be permanent cure, cell at early embryonic stage. ADA is introduced into the cell at early stage, embryonic stage. Permanent cure. Molecular diagnosis. Molecular diagnosis. You know that for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional method of diagnosis, serum and urine analysis, etc., early detection is not possible. Molecular diagnosis. You know that for effective treatment of disease, early diagnosis and understanding its pathophysiology is very important. Effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional method of diagnosis, serum and urine analysis, etc. Conventional method, diagnosis, serum and urine analysis, etc. Early detection is not possible. Molecular diagnosis, effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. You know that for effective treatment of a disease, early diagnosis, understanding its pathophysiology, effective treatment of the disease, early diagnosis, number one, understanding its pathophysiology, number two, is very important. Okay, understanding its pathophysiology. Effective treatment, early diagnosis and pathophysiology is very important. Using conventional method of diagnosis, serum and urine analysis, etc., early detection. Okay, conventional method of diagnosis, serum and urine analysis, early detection is not possible. Conventional method of diagnosis, serum and urine analysis, etc., early detection is not possible. Recombinant DNA technology, polymerase chain reaction (PCR) and enzyme-linked immunosorbent assay (ELISA) are some of the techniques that serves the purpose of early diagnosis. Recombinant DNA technology. Number one, recombinant DNA technology, PCR, polymerase chain reaction, and ELISA, enzyme-linked immunosorbent assay, are some of the techniques that serves the purpose of early detection. Recombinant DNA technology, PCR, polymerase chain reaction, and ELISA, enzyme-linked immunosorbent assay. Recombinant DNA technology, polymerase chain reaction (PCR) and enzyme-linked immunosorbent assay (ELISA) are some of the techniques that serves the purpose of early diagnosis. You know that for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional method of diagnosis, serum and urine analysis, etc., early detection is not possible. Recombinant DNA technology, polymerase chain reaction (PCR), recombinant DNA technology, polymerase chain reaction (PCR), and enzyme-linked immunosorbent assay (ELISA) are some of the techniques that serves the purpose of early diagnosis. 3. rDNA, PCR, and ELISA. Presence of pathogens, bacteria, virus, etc. is normally suspected only when pathogen has produced a disease symptoms. Presence of pathogen, normally suspected only bacteria virus is normally suspected only when pathogen has produced disease symptoms. By this time, the concentration of pathogen is already very high in the body. However, very low concentration of bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PCR. Can be detected by amplification of their nucleic acid by PCR. Can you explain how PCR can detect a very low amount of DNA? Is now routinely used to detect HIV in suspected AIDS patient. HIV is suspected in AIDS patient. It is being used to detect mutations in genes. It is being used to detect mutations in genes in suspected cancer patient. Suspected cancer patient. It is powerful technique. It is a powerful technique to identify many other genetic disorder. It is a powerful technique to identify many other genetic disorder. Okay. Presence of pathogen, bacteria, virus, etc. is normally suspected bacteria virus is normally suspected only when the pathogen had produced a disease symptoms. By this time, the concentration of pathogen is already very high in the body. However, very low concentration of the bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid. Can be detected by amplification of their nucleic acid by PCR. PCR is now routinely used. PCR is now routinely used to detect HIV in suspected patients. AIDS mutation. PCR is PCR is a powerful technique to identify many other genetic disorder. PCR help us to detect HIV in suspected AIDS patient. It is being used to detect mutation in gene in suspected cancer patient. It is being used to detect mutation in gene in suspected cancer patient too. PCR is now routinely used to detect HIV in suspected AIDS patient, detect mutation in gene in suspected cancer patient. Identify powerful technique to identify many other genetic disorder. Identify powerful technique. A single stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complete A single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize. A single-stranded DNA or RNA A single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. Autoradiography. The clone having the mutated gene will hence on the not appear on the photographic film because the probe will not have complementarity with the mutated gene. A single-stranded DNA or RNA tagged a single-stranded DNA or RNA tagged with a radioactive molecule probe. Single-stranded DNA or RNA tagged with radioactive molecule probe. Radioactive molecule is allowed to hybridize to its complementary DNA in a clone in a clone of cells followed by detection using autoradiography. Astaghfirullah. In a clone of cells followed by detection using autoradiography. Detection using auto. First slot is done, okay? Those who want to take break ले सकते हैं, लेकिन ना मैं थोड़ा कंप्लीट करूँगी ये पैराग्राफ कंप्लीट करके ना आई टेक ब्रेक, ओके? स्टडी में मुझे खुद okay, same punch. Single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementarity DNA in a clone of cells, followed by detection using autoradiography. Using autoradiography. A single-stranded DNA or RNA tagged with a radioactive molecule is allowed to hybridize to its complementary DNA in a clone of cell, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film. The clone having mutated gene will not appear on the photographic film because the probe will not have complementarity with the mutated gene. ELISA is based on the of antigen. ELISA is based on the principle of antigen antibody interaction. Antigen antibody infection by pathogen. Infection by pathogen can be detected by presence of antigens, protein, glycoprotein, etc., or by detecting the antibodies synthesized against the pathogen. ELISA is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigens, protein or glycoproteins, or by detecting the antibody synthesized against the pathogen. A single strand, okay. Recombinant DNA technology, polymerase chain reaction, and enzyme-linked immunosorbent assay. ELISA. A single-stranded DNA or RNA tagged with a radioactive molecule probe. A single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by the detection using autoradiography. The clone having the mutated gene will not appear on the photographic film because the probe will not have complementarity with the mutated gene. ELISA based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigen, protein, glycoprotein, etc. Can be detected by the presence of antigens, protein, glycoprotein, etc., or by detecting the antibodies synthesized against the pathogen. Okay. अभी भी 2% बचा है. 5 मिनट का ब्रेक लेते हैं ना. श्री, are you there? Others, are you there? निधि आर यू देयर? कोई नहीं है क्या? 5 मिनट का ब्रेक लेंगे, ओके? नेक्स्ट स्लॉट स्टार्ट करने से पहले ब्रेक से पहले ही स्टार्ट करते हैं, ओके? बाद में लेंगे, ब्रेक लेने का दिल करे तो तब लेंगे, ओके? Transgenic animals. Animals have had their DNA manipulated to possess and express an extra foreign gene are also known as transgenic animals. Animals that have had their DNA manipulated. Animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Animals that have had their DNA manipulated to possess and express and express extra to possess and express an extra foreign bits, pigs, sheep, cows, and fish have been produced. Transgenic rats, rabbits, pigs, sheep, cows, and fish have been produced. Animals that have had their DNA or engineered are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, and fish. Transgenic rats, rabbits, pigs, sheep, cows, and fish have been produced. Although over 95% of all existing transgenic animals are mice, 95% of all existing transgenic animals are mice. These animals being produced. How can man benefit from such modification? Normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated. Specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example: study of complex factors involved in growth, such as insulin like growth factor, by introducing genes from other species that alter the formation of this factor, and studying the biological effects that result. Information is obtained about the biological role of the factor in the body. Study of disease. Many transgenic animals are designed to increase our understanding. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specially made to serve as models for human disease, so that the investigation of new treatment for disease is made possible. Today, transgenic model exists for many human disease, such as cancer, cystic fibrosis, rheumatoid arthritis, and rheumatoid arthritis, cystic fibrosis, and cancer. Transgenic model exists for many human disease: cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer. Cancer, cystic fibrosis, Alzheimer, and rheumatoid arthritis. Study of disease. Normal physiology and development. Biological products. I think I'll be fine. Biological products. Medicines required to treat certain human disease can not control, it's contain. Medicines required to treat certain human disease can contain biological products. But such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of portion of DNA or genes which code for particular products, such as human protein alpha-1 antitrypsin used to treat emphysema. Okay, human protein alpha-1 antitrypsin used to treat emphysema. Similar attempt after being made for treatment of phenylketonuria. PKU. The first transgenic cow, Rose. First transgenic cow, Rose, produced human protein enriched milk, 2.4 grams per liter. The milk contained the human alpha lactalbumin, and was nutritionally a more balanced product for human babies than natural cow milk. First transgenic cow, Rose, produced human protein enriched milk 2 to 4 grams per liter. Protein enriched milk 2 to 4 grams per liter. Protein enriched milk, human protein enriched milk 2.4 grams per liter. 2.4 grams of human protein enriched milk per liter. Human protein 2.4 grams enriched milk per liter. The milk contained human alpha lactalbumin, human alpha lactalbumin, and was nutritionally a more balanced product for human babies than ये वाला PYQ था First transgenic cow, Rose. Transgenic cow, first Rose, human protein enriched milk 2.4 gram per liter. 2.4 gram per liter. Human alpha lactalbumin. Vaccine safety. Transgenic mice are being developed for use of testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccines. Testing on safety of vaccines. Testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of monkey. If successful and found to be more reliable, they could replace the vaccine. The successful and found to be reliable, they could replace the use of monkey. They could replace the use of monkey to test the safety of batches of the vaccine. Chemical safety testing. This is known as toxicity safety testing. Chemical safety testing. Toxicity safety testing. The procedure is the same as The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to the toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain results in less time. Ethical issues. The manipulation of living organisms by the human race cannot go on any further without regulation. Some ethical standards are required to evaluate the morality of all human activities that might help or harm living organisms. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organism can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organizations such as GEAC, Genetic Engineering Approval Committee. GEAC, Genetic Engineering Approval Committee, which will make decision regarding the validity of GM, genetically modified research, and the safety of introducing GM organism for public services. The modification uses of living organism for public service. The modification uses of living organism for public service as food and medicine source. The modification uses of living organism for public service as food and medicine source with patent granted for the same. There is growing public anger that certain companies are being granted patents for product and technologies that make use of the genetic materials, plants, and other biological resources that have long been identified, developed, and used by farmers and indigenous people of the specific region country. Rice is an important food grain, the presence of which goes back thousands of years in Asia's agricultural history. There are an estimated 100,000 lakh 2 lakh varieties of rice in India alone. 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati. 27 documented varieties of basmatis are grown in India. Documented varieties. Okay, there is reference to basmati in ancient text, folklore, and poetry, as it has been grown for centuries. In 1997, an American company got patent right on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad. The new variety of basmati had actually been derived from Indian farmers' varieties. Indian basmati was crossed Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or novelty. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent use as products and process based on Indian traditional herbal medicine. Also been made to patent uses products and processes based on Indian traditional herbal medicines. Example, turmeric, neem. Vigilant, and we do not immediately counter these patent applications. Other countries, individual may encash on our rich legacy, and we may not be able to do anything about it. Biopiracy is the term used to refer the use of biopiracy is the Biopiracy, the term used to refer the use of bioresource by multinational companies and other organizations without proper authorization from the country and people. Use of bioresource by the multinational companies and other organizations without proper authorization from the countries, people concerned without compensatory payment. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity. In contrast, the developing and underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort, and expenditure during the commercialization. There has been growing realization of the injustice in adequate compensation and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment of the Indian Patent Bill that takes such issues into consideration, including patent term emergency and research and development initiative. हाँ तो कर लिया शायद थोड़ा बहुत, अभी तो नींद ही आ रहा है. What are we supposed to do? चैप्टर कंप्लीट हो गया है वैसे मेरा. आई थिंक मेरे कोई फ्रेंड भी नहीं है. आदर्श आर यू देयर? आदित्य आर यू देयर? निधि आर यू देयर? सब लोग चले गए हैं तो मैं भी चली जाती हूँ. ओके, कोई है जो पढ़ रहा है? ओके, आदित्य इज देयर। पहले क्यों नहीं बोला? मुझे ऐसा फील हुआ कि मैं अकेली पढ़ रही हूँ। कब तक पढ़ोगे आदित्य? टेस्ट दे देती हूँ ना एक। आदित्य कब तक पढ़ोगे? I'm going to sleep now. If you are tired, you should rest as well. नहीं, मुझे क्वेश्चन भी प्रैक्टिस करना है ना। अभी चैप्टर कंप्लीट किया है, क्वेश्चन प्रैक्टिस नहीं करूँगी तो कल फिर सब कुछ भूल जाऊँगी। 11:30 भी नहीं हुआ है। आई कांट बिलीव शुभम लोग सो गए आज। कैसे सो गए वो लोग आज इतनी जल्दी? बायोलॉजी। बायोटेक्नोलॉजी एंड इट्स एप्लीकेशन ही था ना ये। Transgenic plants are the one generated by introducing foreign DNA into the cell and regenerating a plant from that cell. Produced after grown in artificial medium after hybridization in the field. Produced by a somatic embryo and in artificial medium. Transgenic plants are the ones generated by introducing foreign DNA into cell and regenerating a plant from that cell. Extra foreign genes are known as transgenic animals. Transgenic plants are the one generated by introducing foreign DNA and regenerating a plant from that cell. The organisms that carry foreign genes, a combination of genetic material obtained through the use of modern biotechnology, are called mutant genotype transgenic recombinant, or the organism that carries foreign gene. Obviously recombinant, then transgenic. An organism that contains a foreign gene is called transgenic. An organism that contains a foreign gene. Transgenic. Okay. Possesses and express. Okay, DNA manipulate to possess extra foreign gene are known as transgenic animals. The organisms that carry foreign genes, a combination of genetic material obtained through the use of modern biotechnology, are called transgenic. Okay. Alpha 1 antitrypsin, emphysema, eco 3. देखो cry 1 AB, try 1 AC, corn borer Bico 4, Seco, cotton bollworm, Seco 1, Bico 4, Seco 1. Enzyme replacement therapy, injection, ADA deficiency. When genes was introduced in the first which gene was introduced in the first transgenic cow? Human alpha lacked albumin, alpha 1 antitrypsin. Okay, first one is alpha human alpha lacked albumin. Bt toxin protein crystal present in bacterium Bacillus thuringiensis do not kill the bacteria themselves because bacteria are resistant. Toxins are in inactive prototoxins in bacteria. Correct. Which statement is incorrect about gene therapy? Lymphocytes from patients' blood are cultured. A functional ADA cDNA is introduced into this lymphocyte. Lymphocytes are then introduced into the body of the patient. Patient does not require periodic infusion. Genetically engineered lymphocyte incorrect. Which is correct regarding genetically engineered insulin using E. coli? Difficulty to purify, obtain in large unlimited quantities, possibility of transmission of animals disease, insulin obtained varies in chemical structure. Option B, obtained in large unlimited quantities. And the application of biotechnology include therapeutics. The application of biotechnology include therapeutics, diagnosis, GM, genetically modified crops for agriculture, processed food, bioremediations, waste treatment, energy production. All of this. Gene therapy, humulin probe and ELISA probe. Gene therapy, humulin, ELISA. Type test, diagnostic test, ELISA, DECO1. A single-stranded DNA or RNA tagged with a radioactive molecule. Probe, a single-stranded DNA or RNA tagged with a radioactive molecule. SECO2, DECO3, SECO2, DECO3, SECO2, DECO3. SECO2, DECO3. Humulin. Effort to fix functional gene. Gene therapy. Effort to fix functional gene. Effort to fix functional gene. Gene therapy. ECO1. Effort to gene therapy. Effort to fix functional gene. ELISA. Diabetes. Diagnostic test. Okay, go. ELISA is diagnostic test. Yes. Humulin. Diabetes. Bico for humulin diabetes. Humulin diabetes. I thought Nidhi and Adarsh you guys went. मैंने कितनी बार बुलाया भी था. कहाँ जाते हो तुम लोग? Gene therapy. Effort to fix functional gene. Humulin diabetes. Bt brinjal is an example of transgenic crop. In this, Bt refers to Bacillus thuringiensis. Which gene does not appear in? मुझे ना पकौड़ा खाने का दिल कर रहा है लेकिन मैं छीला बना के खाऊं क्या? वैसे मेरे पास राइस भी है लेकिन मुझे छीला ही खाने का दिल कर रहा है आज. Which gene does not appear in photographic film in autoradiography. Housekeeping gene, structural gene, mutated gene. Mutated gene does not appear in photographic film in autoradiography. Bacteria genetically engineered to express a gene from a plant will synthesize a protein with the same sequence of amino acid as in the plant, and therefore the protein will have the same structure and function as in plant. Engineered to express a gene from a plant will synthesize a protein with essentially the same sequence of amino acid as in the plant with different relating to different codon vowel rule between prokaryotes and eukaryotes. Now, okay, bacteria genetically engineered transcriptional processes does not occur in it. Gene from a plant. Express a gene from plants, blood, fungi, or slightly different approaches used. Which of the following statement which of the following statement are correct regarding the process of RNA interference? This is used to prevent the infestation of protozoans. It takes place in some eukaryotes and all prokaryote organisms as a method of cellular defense. 2 is false. जहाँ जहाँ 2 है उसको eliminate करो. Correct, incorrect. 3. The method involves silencing of specific mRNA due to the complementary double-stranded RNA molecule. It is novel strategy to produce pest-resistant plant. 4, 3, and 4. Bt corn has been made resistant from the corn borer disease by introduction of the gene Cry1AB, Cry2AB, Cry1AB. Given below are two statements. Bt toxins are insect group specific and coded by a gene Cry1AC. Bt toxin exists in inactive rhodotoxin in Bacillus thuringiensis. However, after ingested by the insect, the inactive rhodotoxin gets converted into active from due to acidic pH of the insect gut. Statement 2 is incorrect. The site of production of ADA in the body is erythrocytes, lymphocytes, blood plasma, osteocytes. Lymphocytes. Transgenic plants are produced by क्यों क्या किया आज शुभम Transgenic plants are produced by inducting gene mutation, arresting spindle fiber formation, deleting chromosomes, introducing foreign genes. Transgenic plants are produced by introducing foreign genes. वो कहाँ गया नहीं थी तुम्हारा तो मतलब कि बैडमिंटन में जीता है ना किसके साथ था वैसे मैच all the biotechnological application in order to increase food production except apiculture, agrochemical based agriculture, organic farming, genetically engineered crop based apiculture, beekeeping. A patent is granted for an invention, an improvement in an earlier invention, a process of generating a product, strains of microorganism. Process of generating a product. American company got patent. Right on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati. The new variety of basmati had actually been derived from the Indian farmer's variety. It was crossed with semi-dwarf varieties and claimed as an invention. Or novelty. The patent extend to functional equivalent, implying that other people saying basmati rice could be restricted by the patent. Alhamdulillah, मैं तो डर ही जाऊंगी. Process of generating a product. 1 तो यही है, 1, 2 and 3. Improvement in earlier invention and process of generating a product. इतनी जल्दी आने लगी नींद. कॉफ़ी बना के आती हूँ. ओके निधि. चाय यहाँ की दुकान पे कॉफ़ी ख़त्म हो गया है. दोनों ही दुकान पे. मैं तो चाय पी रही हूँ कब से. शुभम मैच किसके साथ था? An invention, an improvement in earlier invention. Invention and improvement in earlier invention, process of generating products, strains of microorganisms. Process of generating a product, an improvement in earlier invention, an invention. Permanent cure for ADA deficiency is ADA gene introduced in early embryonic stage. ADA gene introduced in early embryonic stage. Lymphocyte of SCID. Patients are treated by introduction of DAS. These treated lymphocytes are subsequently returned to the patient. Lymphocytes of SCID patient are treated by the introduction of functional ADA RNA, functional ADA cDNA, SCID. ADA, SCID. Functional ADA cDNA. Techniques used to detect the DNA in a clone is polymerase chain reaction, gel electrophoresis. Techniques used to detect the DNA in a clone is chromatography, autoradiography. Polymerase chain reaction. Technique used to detect the DNA in clone is PCR, gel chromatography, autoradiography. DNA clone, okay, autoradiography. Techniques used to detect the DNA in clone is a single-stranded DNA or RNA tagged with a radioactive molecule probe. Okay, a single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybrid to its complementary DNA. A single-stranded DNA or RNA tagged with a radioactive molecule is allowed to hybride to its complementary DNA in a clone of cell. Astaghfirullah. In a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will not appear on the photographic film because the probe will not have complementarity with the mutated gene. Technique used to detect the DNA in clone: autoradiography. When DNA is transcribed into mRNA, usually the mRNA remains single-stranded, but in some cases, an RNA can be made that is complementary to the mRNA. This is called antisense reverse transcription, and its main function is to block gene expression, amplify mRNA, enhance translation. Block gene expression. When DNA is transcribed into mRNA, usually the messenger RNA remains single-stranded, but in some cases the RNA can be made and it's complementary to the mRNA. In cases, an RNA can be made that is complementary to the mRNA. This is called a dash antisense, and its main function is to block gene expression. This is called antisense RNA. Antisense RNA. You will be fine, Ima. Antisense RNA. Who knows? Silencing of a gene could be achieved through the use of short interfering RNAi, antisense RNA, both A and B. Short interfering RNAi, RNA interference. Silencing of gene could be achieved through the use of antisense RNA and short interfering RNAi. Two bacteria found to be very useful in genetic engineering experiment are Agrobacterium and E. coli. Agrobacterium, I think वेक्टर था ना, मैं भूल गई. वेक्टर था, हाँ, nematode specific gene. Cryoprotein are named so because they are crystal protein. Cryoproteins are soluble in acid environment of insect midgut and then release toxic fragment after proteolytic action. Very basic होता है. Assertion is true but reason is false. Given figure represents the maturation of proinsulin into insulin. Okay. Identify the product A. Where is product A? Okay, this is cDNA. Polypeptide chain A, polypeptide chain B, polypeptide chain C. Option C. Transgenic animals are generally produced for all the following needs except testing for chemical safety, testing for vaccine safety, stimulation of pathogenicity, production of pharmacologically important proteins. Transgenic animals are produced for all of the following needs except stimulation of pathogenicity, production of pharmacologically important protein. अच्छा यही option C, stimulate of pathogenicity. Which of the following can help in diagnosis of genetic disorder? ELISA, genetic disorder, ABO blood group, PCR and MR. ELISA and PCR है ना? हाँ ना, PCR है, PCR. not ELISA, okay? PCR, detected by amplification of the nucleic acid by PCR. HIV, autoimmune. It is being used to detect mutation in genes suspected cancer patient. Genetic disorder. Which of the following helps in diagnosis of genetic disorder? PCR. PCR helps in diagnosis of genetic disorder, okay? One of the advantages of developing transgenic mice is that it is very useful to study vaccine safety. It is in producing new varieties of mice in developing a showpiece example. अच्छा ये first का ये to study vaccine safety. Study the following statement regarding organic farming and select the correct ones. It utilizes genetically modified crop like BT cottons. It uses the naturally produced inputs like compost and biofertilizers. Correct. Option 2. You correct. Okay. You think B is eliminated. It does not use pesticides and urea. Organic farming 3. Okay, 2 and 3. Choro. 2 and 3. Okay. 2, 3 and 4. It produces vegetables rich in vitamins and minerals. 2, 3, 4. 2 and 3. In organic farming, farmers use manures, biofertilizers, pesticides, and biocontrol agents to increase the crop production instead of using artificial fertilizers and pesticides. It does not utilize genetic engineering. The transgenic animals are those which have foreign DNA in all its cell, foreign DNA in some of its cell, foreign RNA in all of its cell. Foreign DNA in all its cell. Transgenic animals are those which have foreign DNA in all its cell. Nowadays, it is possible to detect the mutated gene causing cancer by allowing radioactive probe to hybridize its complementary DNA in a clone of cell, followed by detection using autoradiography because a mutated gene clearly does not appear on a photographic film, as the probe has no complementarity with it. Has no complementarity with it. Okay, has no complementarity. Not appear on the because the probe will not have complementarity with the mutated gene. Not have complementarity with the mutated gene. 35 तक करते हैं ना. Which step has been taken by government of India to cater to the requirement of patent terms and other emergency provisions in this regard? Biopiracy Act, Indian Patent Bill. Okay. Which of the following statement is not correct? Insulin, not correct. Insulin used for diabetic patients was earlier extracted from pancreas of slaughtered cattle and pigs, which was more efficient than the genetically engineered. First का ही गलत है. PCR technique is applied to detect HIV in suspected AIDS patient and to detect mutation in genes in suspected cancer patient. Correct. Bone marrow transplantation requires periodic infusion of genetically engineered lymphocytes in ADA deficient patient. Adenosine deficiency deaminase. Bioremediation is one of the application of biotechnology. Okay, got it. हो गया. टेस्ट दे दिया यार. मुझे नींद. ओके निधि तुम वापस आ गई हो क्या कॉफी लेके? अस्तगफिरुल्लाह। हाँ, इतना सिली क्वेश्चन कौन पूछता है, गुस्सा ही आ जाता है मैंने। सोए की क्या करी नहीं थी? तुम बोलो कब तक? मुझे तुम्हें तो अभी भी सो सकती हूँ क्योंकि मैंने कुछ चाय कॉफ़ी कुछ नहीं पिया है। कब तक पढ़ना है? बोलो ना निधि। क्या पता है? नहीं पता है। ट्वेल्व होने ही वाला है। अभी लेट सोऊंगी, कल लेट उठूंगी, फिर लेट। तो जल्दी सो जाओगी? मैं तो नाइट आउल हूँ, सुबह नहीं पढ़ सकती। मैं जितना पढ़ूँ अभी पढ़ सकती हूँ। 2 AM मैक्स। ओके तो ठीक है, 2 AM तक ही पढ़ते हैं। ओके मैं फेस वॉश करके आई। ओके मैं तो कॉफ़ी पीता हूँ फिर स्टडी। ओके कितना ठंड लगा ऑल ऑफ ए सडन। अबे कंजूस, वो ऐड ऑन कर सकता है तू? मुझे लगा कि तू मर गया, सो गया। फिर थैंकफुली कहाँ से नींद आ गई टाइम पे, वरना मैं बायोटेक का वो सेकंड चैप्टर भी कंप्लीट नहीं कर पाती थी। हाँ मुझे भी पता है नहीं मरोगे, मैंने बोला ना, मुझे लगा कि तुम सो गए। मैं इतनी शॉर्ट नहीं हूँ, ये टेबल मेरे लिए काफ़ी छोटा पड़ जाएगा। आदर्श आदर्श के एक वाले चैनल पे ना उसका प्रोफाइल पिक पे काफका का है और दूसरा वाला ये सॉन्ग वाले में ना उसने चेंज करके वो रख दिया, नाकाता रख दिया। इतना अच्छा लगा क्या काफका ऑन द शोर? बिल्लू भाग गया, उसके बारे में मत, मुझे बुरा लग जाएगा। मुझे यहाँ पर ना उसे लैप में रखते रहना ना, मुझे गुस्सा आया ना, तो मैंने उसको निकाल के यहाँ पर रख दिया था। फिर उसको ना शायद बुरा लगा, इसलिए वो बाहर चला गया था। फिर अम्मा ने उसको भगा दिया कहीं और। जीत पे गई। वाह निधि, तुम ऐसी चीज़ भी करती हो, शायरी भी बोलती हो। वो बहुत फेक लग रहा है विष्णु। आदर्श ने क्या लिखा था, किसी ने देखा? मैंने नहीं देखा। ह्यूमन फिजियो, हाँ एक बार कर तो रखा है, लेकिन रिवीजन करना पड़ेगा। ह्यूमन फिजियो ही पूछा ना, प्लांट फिजियो भी कंप्लीट किया है वैसे, लेकिन वो भी रिवीजन करना पड़ेगा। मैं सब कुछ भूल जाती हूँ। मैंने बायोटेक ना ये कुछ दिन पहले ही किया था, मैं वो भी भूल चुकी हूँ। ये उतना तो नहीं, पर समथिंग स्टार्टेड विद इट। ओह, मेरा तो बायो ही रिवीजन नहीं हो पा रहा है भाई। क्या मतलब केवल बायो ही रिवीजन होगा? बायो भी नहीं हो पा रहा है। नहीं है तो एक दो डिफरेंट डिफरेंट। हमारे शुभम भी तो नेट रिफ्रेश हो गया। इट डजंट मैटर तुम किस चीज़ के लिए पढ़ाई कर रहे हो, इतना ही मैटर करता है कि तुम पढ़ाई कर रहे हो। क्योंकि हम लोगों को ना सिर्फ इस चैनल में स्टूडेंट्स चाहिए। एवोल्यूशन। कॉकरोच जैसे गंदा छापा कर लिया। Loss of biodiversity. देखते हैं। ओके, कोशिश करेंगे। ओके, वो भी पढ़ेंगे। मजे के लिए थोड़ी पढ़ रहे हैं भाई। आर यू देयर एसबी? मेरे सामने बिल्कुल तुम्हारा वो विदाउट स्पेक्स वाला पिक्चर खुल गया। आई होप निधि इधर होगी ना, तो मैं निधि को दिखा दूँ फ़ोन से पकड़ के वो वाला पिक। मजाक कर रही हूँ, वो पिक नहीं है मेरे पास। मैंने कब का डिलीट कर दिया था वो। 2 सेकंड में भाई ने पैंट गीला कर लिया डर के मारे। छोड़ दो ये। Biodiversity and conservation. Biodiversity and conservation, okay? Good luck, Shubham. Biodiversity and conservation. If an alien from a distant galaxy were to visit our planet Earth, the first thing that would amaze and baffle him would most probably be the enormous diversity of the life that he would encounter. Even for humans, the rich variety of living organisms with which they share this planet never ceases to astonish and fascinate us. If an alien from a distant galaxy were to visit our planet Earth, the first thing that would amaze and baffle him would most probably be the enormous diversity of the life that he would encounter. Even for humans, the rich variety of living organisms with which they share this planet never ceases to astonish and fascinate us. The common man would find it hard to believe that there are more than 3 lakh, 1 tens, 100,000, 10,000, 1 tens, 100,000, 10,000 lakh, 3 lakh. मेरे पास इतना होता ना 3 lakh 20,000 species ants and 3 lakh species of beetles, 28,000 species of fishes, 28,000 species of fishes and nearly 20,000 species of orchid. Okay, 20,000 ants and or 8,000 species of fish and fishes and 3 lakh 3 lakh सबसे ज़्यादा 3 lakh species of beetles, 20,000 species of ants, 20,000 species of orchid, 3 lakh species of beetle and 28,000 species of fish. 20,000 ants and orchid, ORCHIDs, orchids species, 28,000 fishes, FISHESH, fishes and 3 lakh beetles. 3 lakh beetles, okay? Ecologists and evolutionary biologists have been trying to understand the significance of such diversity by asking important questions. Why are there so many species? Did such throughout It's okay, ma. मुझे वो सोच के हंसी आ रही है, लेकिन इट्स ओके है ना? मैं क्यों पॉज हो गई ऐसे करके? There are why are there so many species? Did such great diversity exist throughout Earth's history? How did this diversification come about? How and why is this diversity important to the biosphere? Commensalism and amensalism. Amensalism दुईटा नेगेटिभ, commensalism एउटा पोजिटिभ एउटा नेगेटिभ. एउटा पोजिटिभ एउटा जिरो commensalism, एउटा नेगेटिभ एउटा जिरो amensalism. Negative and neutral, amensalism, positive and neutral, commensalism, parasitism, positive and negative, predation, positive negative, competition, negative, mutualism, positive, competition, negative, both has negative, both the species will get negative effect. Competition, mutualism, positive, predation, positive, negative, parasitism, commensalism, amensalism. I think 1, 2. The evolution. Why are there so many species? Did such great diversity exist throughout Earth's history? How did the diversification come about? How and why is this diversity important to the biosphere? Would it function any different if the diversity was much less? How do human benefits from the diversity of lives? Astaghfirullah. Biodiversity. In our biosphere, immense diversity or heterogeneity. Heterogeneity. In our biosphere, immense diversity or heterogeneity exists not only at the species level, but at all levels of biological organization, ranging from macromolecules within cell to biomes. In our biosphere, immense diversity or heterogeneity exists not only in the species level. Diversity exists not only in the species level, but at the levels of biological organization, ranging from macromolecules within cells to biomes. Biodiversity is the term popularized by the sociobiologist. Biodiversity is the term popularized by sociobiologist Edward Wilson to describe the combined diversity at all levels of biological organization. Biodiversity. Sociobiologist Edward Wilson described combined diversity. Combined diversity at all the level of biological organization. मुझे काफ़ी ख़राब लगा था तब, लेकिन क्या ही कर सकते हैं. Biological organization. Okay. The most important of them are biodiversity is the term popularized by the sociobiologist Edward Wilson. Biodiversity, Edward Wilson, biodiversity. 20,000 species of orchids, 28,000 species of fishes, and 3 lakh species of beetles. Biodiversity, sociobiologist Edward Wilson. Combined diversity at all level of biological organization. The most important of them are biological organization, genetic diversity, species diversity, and ecological diversity. Biodiversity, genetic diversity, ecological diversity, and species diversity. Genetic diversity, species, GSC, ESG, SEG. Gen, species, eco. Gen, species. Okay, gen. Genie speak on ecology. Genie, G-A-N-Y, Genie speak on ecology. Genie speak this is trick. Gen, genetic diversity, gen, speak. Species diversity, genetic diversity, species diversity, and one last, ecological diversity. Genetic diversity, species diversity, and ecological diversity. Genetic diversity. A single species might shows high diversity at genetic level over its distribution range. The genetic variation shows by the medical plant Rauwolfia vomitoria, growing in different Himalayan range, might be in terms of the potency and concentration of the active chemicals reserpine that the plant produce. India has more than 50,000 genetically different strains of rice. 50,000 genetically different strains of rice and 1000 varieties of mango of rice and 1000 varieties of mango. Genetic diversity. A single species might shows high diversity. Single species high diversity shows high diversity at genetic level. Single species shows high diversity at genetic level. Over its distribution range, over its distributional range, the genetic variation shows by the medicinal plant. Okay, medicinal plant Wolfia vomitoria growing in different Himalayan ranges might be in terms of potency and concentration of active chemical. Might be different Himalayan ranges might be in terms of potency and concentration of active chemicals. Reserpine. Which chemical? Active chemical reserpine. Reserpine that the plant produce. India has more than 50,000 genetically different strains of rice. 50,000 genetically different strains of rice and 1000 varieties of mango. Genetic diversity. Genetic diversity. Genetic diversity भनेको के हो? Genetic diversity में species, one species, multiple variation. One species, multiple variation. Multiple variation. Variation. Multiple variation. आज तक फिर होला, I'll be fine है ना? I guess so. एउटा species हुन्छ. One species, multiple variation. One species, multiple variation. Variation. Okay, diverse. High diversity. के भन्छ यसलाई? High diversity. Diversity. High diversity. A single species might shows high diversity at genetic level. High diversity at genetic level. Genetic level. Genetic diversity भनेको एउटा species को high diversity हुन्छ genetic level मा. Okay. Example. Medicinal CINL. Medicinal plant. Vomiteria. Wolfia. Raw RAU. Raw Wolfia. Raw Wolfia. Vomiteria. Vomiteria. RAU. Raw Wolfia. Raw Wolfia. Wolfia. Vomitoria. 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 Astaghfirullah. Astaghfirullah. Raw Wolfia. I think I like his personality. लेकिन all of a sudden कैसे याद आ गया पढ़ाई के बीच में? DIFFERENT. Different in terms of chemical, active chemical, concentration of active chemical called reserpine. Okay, medicinal plant raw wolfia. Okay, in terms of potency and potency, potency and concentration of active chemical. Concentration of active chemical reserpine. Active chemical. Okay, उसको raw via vomitoria. Okay. 50,000 strains. 50,000 different strains. Different STRA strains of rice and 1,000 varieties of mango. Genetic diversity. Genetic diversity. एउटा स्पिसिसको जेनेटिक डिफरेन्ट जेनेटिक भेरिएसन हुन सक्छ, जसलाई के भन्छ जेनेटिक डाइभर्सिटी. Species diversity. The diversity at the species level. For example, the Western Ghats have a greater amphibian species diversity than Eastern Ghats. Okay, Western Ghats have greater Western Ghats. The diversity at species level. Diversity at species. Western Ghats have greater amphibian species diversity than Eastern Ghat. Okay, diversity at species level. For example, Western Ghat ma ke species diversity species diversity WESTERN Western Ghat GHAT घाट में ज़्यादा डाइवर्सिटी हुन्छ EASTERN Eastern घाट में हुँदा. ये क्यों ऐसा पॉज हो रहा है बार बार? देखो फिर से हुआ ना. लेट मी चेक. नहीं, वो वाईफाई से ही कनेक्टेड है. Western Ghat has more species. Diversity. Amphibians than Eastern Ghat. Okay, Western Ghat have a greater amphibian species diversity than the Eastern Ghat. Ecological diversity at the ecosystem level. मुझे खुद की है ये देख के ना कितना अच्छा लगा. Ecosystem diversity at the ecosystem level. Ecological diversity at the ecosystem level. India, for instance, with the desert, rainforest, mangrove, coral reef, wetland, estuaries, and alpine meadows, has a greater ecosystem diversity. At the ecosystem level, India, for instance, with its desert, rainforest, mangroves, coral reefs, इसमें से ना आधा तो मुझे पता भी नहीं है. Wetlands, estuaries, and alpine meadows has a greater ecosystem diversity than Scandinavian country like Norway. Scandinavian country like Norway. For instance, with desert, rainforest, mangrove, coral reef, wetlands, estuaries, and alpine meadow has greater ecosystem diversity than Scandinavian country like Norway. Mangrove. Coral reef, wetland, estuaries. Let's see, Scandinavian. Is a submerged of Northern Europe. Northern Europe with a strong historical culture and linguistic ties between the constituent people. Norway. Belonging or relating to a group Northern European countries that includes Denmark, Norway, and Sweden. Sub-region of Northern Europe. Ecological diversity at the ecosystem level. Ecological diversity at ecosystem level. India, for instance, with its desert, rainforest, mangrove, coral reefs, wetlands, estuaries, and alpine meadows, has a greater ecosystem diversity than a Scandinavian country like Norway. It has taken millions of years of evolution. It has taken millions of years of evolution to accumulate this rich diversity in nature. But we could lose all that wealth in less than 2 centuries. If the present rate of species losses continues. Biodiversity and its conservation are now vital environmental issue. Biodiversity and conservation are now important environmental. Biodiversity and its conservation is now important vital environmental issues of international concern as more and more people around the world begins to realize the critical importance of biodiversity for our survival and well-being on this planet. Biodiversity and its conservation, vital environmental issue. Biodiversity and its conservation. Biodiversity and its conservation. Genetic diversity, species diversity. Amphibians species diversity in Western Ghat is more than Eastern Ghat. एम्फीबियंस की बात कर रहे हैं हम लोग. कोरल रीफ्स, वेटलैंड्स, mangroves, rainforests, desert, estuaries. Ecosystem level, India has greater ecosystem diversity than Norway. First is genetic diversity. Example, raw wolfia. Rhizopin, okay. Vomitaria. Raw wolfia, vomitorian, vomitoria. Biodiversity and its conservation, vital environmental issue. How many species are there on Earth and how many in India? Since there are published records of all the species discovered and named, we know how many species in all have been recorded so far, but it is not easy to answer the question of how many species there are on Earth according to International Union of Conservation of Nature and Natural Resources, IUCN, International Union of Conservation of Nature and Natural Resources. 2004, the total number of plant and animal species described so far is slightly more than 1.5 million, but conservation of nature and natural resources. 2004, the total number of plant and animal species described is so far is slightly more than 1.5 million, but we have no clear idea of how many species are yet to be discovered and described. Estimates vary widely and many of them are only educated guesses. For many taxonomic groups, species inventories are more complete in temperate than in tropical countries. Species inventories are more complete in temperate than in tropical countries, considering that an overwhelming large population of the species waiting to be discovered are in tropics. Biologists make a statistical comparison of the temperate tropical species richness of an exhaustively studied group of insects and extrapolate this ratio to other group of animals and plants to come up with a gross estimate of the total number of species on Earth. Some extreme estimates, okay, these are just estimates, range from 20 to 50 million, but a more conservative and scientifically sound estimate made by Robert May placed the global species diversity at about 7 million. Estimate made by Robert May. Global species diversity about 7 million. Global species diversity 7 million. ये भाई को अभी तक study loud. I want to know what you are reading. मैं तो यूपीएससी के लिए पढ़ भी नहीं रही हूँ. क्या बोल रहा है ये इंसान. एटलीस्ट इतना तकलीफ तो कर सकता कि चैनल में जाके बाकी वीडियो देखता. नहीं, अज्यूम करना है. भाई साहब आप गलत हो. इंटरनेशनल यूनियन फॉर कंजर्वेशन ऑफ. थैंक यू शुभम. हाँ, इन सबको किक आउट कर देने के लिए. हाइड ही कर देना. What's that? What is SMS, by the way? आई थिंक मुझे ना मेरी सेटिंग्स चेंज करनी चाहिए. International Union for Conservation of Nature and Natural Resources. International Union of Conservation of Nature, Union. International Union for Conservation of Nature and Natural Resources, 2004. International Union for Conservation of Nature and Natural Resources. Total number of plant and animal species described so far is slightly more than 1.5 million. Slightly more than 1.5 million, but we have no clear idea of how many species are yet to be discovered and described. Estimates vary widely and many of them are only educated guesses. For many taxonomic group, species inventories are more complete in temperate than in tropical countries, considering that an overwhelming large portion of the species waiting to be discovered are in the tropics. Biologists make a statistical comparison of the temperate tropical species richness of an exhaustively studied group of insects and extrapolate this ratio to other group of animals and plants to come up with a gross estimate of the total number of species on Earth. Some extreme estimates ranging from 20 to 50 million. Some extreme estimates ranging from 20 to 50 million. 20 million से 50 million. Some extreme estimates ranging from 20 to 50 million, but a more conservative and significantly sound estimate made by some extreme estimates range to 20 to 50 million. Extreme estimates 20 to 50 million, but a more conservative and scientifically sound estimate made by Robert May placed the global species diversity at about 7 million. Robert May, global species 7 million. Robert May, global species diversity. Global species diversity about 7 million. Robert May, global species diversity about 7 million. Let us look at some interesting aspect about Earth's biodiversity based on currently available species inventories. Let's look at some interesting aspect about Earth's biodiversity based on currently available species inventories. More than okay, let us look at some interesting aspects about Earth's biodiversity. Earth's biodiversity based on the currently available species inventories. Let us look at some interesting aspect about Earth's biodiversity based on the currently available species inventories. Let us look at some interesting aspect about Earth's biodiversity based on the currently available species inventories. More than 70% of all the species recorded are animals. More than 70% of species recorded of all the species recorded are animals, while plant including algae, fungi, bryophytes, gymnosperm and angiosperm comprises no more than 22% of the total, including fungi. Okay. Among animals, insects are the most species rich. Among animals, insects are most species rich taxonomic group. Most species rich. Insects are the most species rich taxonomic group, making up more than 70% of the total. Making up more than 70% of the total. That means out of every 10 animal on this planet, 7 are insect. Out of every 10 animals in this planet, 7 are insect. Okay. Again, how do we explain this enormous diversification of insect? The number of fungi species in the world is more than the number of fungi species in this world is more than the combined total. अम्मा उठ के ना ऐसे गाली देती रहती है. The number of fungi species in the world is more than the combined total of the species fishes, amphibians. Of the total species of fishes, amphibians, reptiles and mammals. Of the combined total of the species fishes, amphibians, reptiles, fishes, amphibians, reptiles and mammals. Figure 13.1 biodiversity is depicted showing species number of major taxa representing global biodiversity. Proportionate number of species of major taxa of plant, invertebrates and vertebrates. Invertebrates. Insect, mollusk, insect, other animals, mollusk and crustaceans. Invertebrates. Invertebrates. Insect, then other animal group, mollusk and crustaceans. Mollusks and crustaceans. CEANS. Invertebrates. IOMC. Crush. In out mid crush. Trick. Inside out morning crush. in insects, other animals, then mollusk, MOLLUSCs, mollusk, and crustaceans, CRUSTEANS, crustaceans. Inside, that is insect. Inside out butter, other animals, inside out, mol, M butter, mollusk, crush butter, crush, crustaceans. 40 weights. IUMC. India of my FP. Fish, birds, reptiles. Fish, birds, then reptiles, amphibians, and finally mammals. Okay. Invertebrates. Fish, birds, first fish, then birds, then reptiles, then amphibians, then mammals. Fish, birds, amphibians, and mammals. Fish, birds, amphibians, and fish, birds, reptiles, amphibians, and mammals. Fish, birds, FB. Fish, birds, RAM, are reptiles, A, amphibians, M, mammals. FB, RM, RAM. FB, fish, RAM, room, whatever. Fish, birds, reptiles, amphibians, and mammals. Reptiles, amphibians, and mammals. Fish, birds, reptiles, amphibians, and mammals. Insect or the animal group, mollusk, crustaceans. Insect or the animal group, mollusk and crustaceans. Invertebrates, vertebrates, and finally this one. Organ is fungi. Organ makes fun of losers. That's fungi. Organ. Angiosperm is fungi. Fungi. That is a angiosperm, then fungi. Angiosperm, fungi is fungi. Organ. Algae. Organ. Algae makes mosses. Fun. Ferns. Fern of ferns and allies. Ferns of losers. LOOSERS, losers. Orun से angiosperm. Fungi से fungi. Orun is fungi. That is angiosperm and fungi. Orun makes algae. Makes से mollusk. Mollusk. Ferns and allies. Fifth number ma. Ferns and allies. Fun of losers, lichens, 6 ma. Okay, these are plants. Okay, plants. Orun, angiosperm, then fungi, algae, one, fern and allies, ALLIES, allies. Mosses, 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 mosses. Mosses, ferns. Orun, fungi. Orun makes fun of losers. Lichens, LICHENS, lichens. Angiosperm fungi, algae, mosses, ferns and allies, and lichens. Ferns and allies and lichens. Angiosperm fungi, algae, mosses, ferns and allies, lichens. Angiosperm fungi, algae, mosses, ferns and allies, lichens. More than 70% of all the species recorded are animal, while plant, including algae, fungi, bryophytes, gymnosperm, and angiosperm, comprises no more than 22% of the total. Among animal, insects are the most species-rich taxonomic group, making up more than 70% of the total. That means, okay, more than 70% of all the species recorded are animals, while plant including algae, fungi, bryophytes, gymnosperm, and angiosperm, comprises no more than 22% of the total. 70% animal, 22% plant, 22% plant, along with fungi. Among animal, insects are the most species-rich taxonomic group, make up more than 70% of the total. That means out of 10 animals on this planet, 7 are insect. Again, how do we explain this enormous diversity of insect? The number of fungi species in the world is more than the fishes, more than the combined of fishes, amphibians, reptiles, and mammals. Combined total of species of fishes, amphibians, reptiles, and mammals. Okay. Invertebrates में म आपको कुछ भन्न था इन भनेपछि तिम्रो इन आउट मोल क्रस्टेशियन्स मलबाट मोलस्क MOLLUCS mollusk other animal group insect other animal group mollusk and crustaceans other animal group crustaceans mollusk and insect. Okay, vertebrates FBRAM F fishes FISH fishes, birds, then reptiles, amphibians, and finally mammals. It should be noted that these estimates do not give any figure for prokaryote. It should be noted that these estimates do not give any figure for prokaryotes. It should be noted that these estimates do not give any figure for prokaryote. These estimates do not give any figure for prokaryote. Biologists are not sure about how many prokaryotes species there might be. The problem is that conventional taxonomic methods are not suitable for identifying microbial species. Conventional taxonomic methods are not suitable for identifying microbial species, and many species are simply not culturable. The problem is that conventional taxonomic methods are not suitable. Okay. It should be noted that these estimates do not give any figure for prokaryotes. Biologists are not sure about how many prokaryotic species there might be. The problem is that conventional taxonomic methods are not suitable. For identifying microbial species, and many species are simply not culturable under laboratory conditions. It should be noted that these animals do not give any figure for prokaryotes. Biologists are not sure about how many prokaryotic species there might be. The problem is the conventional taxonomic methods are not suitable for identifying microbial species. The problem is that conventional taxonomic methods are not suitable for identifying microbial species, and conventional methods are not suitable for Many species are simply not culturable under laboratory condition. Not culturable. Okay. Microbial species and many species are simply not culturable under laboratory condition. If we accept biochemical or molecular criteria for delineating species for this group, then their diversity alone might run into millions. If we accept biochemical or molecular criteria, their delineating species for this group, diversity alone might run into millions. Although India has only 2.4% of the world land area, 2.4% of the world's land area, the global species diversity is an okay. Its share of the global species diversity is an impressive 8.1%. 8.1%. Land share 2.4%. Diversity 8.1%. This is what makes our country one of the 12 mega diversity country 12 mega diversity countries of the world. 12 mega diversity countries of the world. Nearly 45,000 species of plant and twice as many of animals have been recorded. 45,000 species of plants and twice as many of animals, 90,000, have been recorded from India. How many living species are actually there waiting to be discovered and named? If we accept May's global estimate, only 22% of the total species have been recorded so far. Applying this proportion to India's diversity figure, we estimate that there are probably more than 1 lakh plant species and more than 3 lakh animal species yet to be discovered. More than 3 lakh animal species yet to be discovered and described. Would we ever be able to complete the inventory of the biological wealth of our country? Would we ever be able to complete the inventory of the biological wealth of our country? Consider the immense trained manpower, taxonomist, and time required to complete the job. The situation appears more hopeless when we realize even before we discover them, nature's biological library is burned. Of all the books stop there. 12 mega diversity country India, 2.4% in world's land area. Global species diversity 8.1%. 8.1% global species diversity. What's wrong with this one? 2.4% of world land area. It shares the global species diversity in an impressive 8.1%. That is what makes our country one of the 12 mega diversity countries of the world. Nearly 45 species of plant and twice as many of animals have been recorded from India. How many living species are actually there waiting to be discovered and named? If we accept Mary's global estimate, 22% of the total species have been recorded so far. 1 lakh plants or 3 lakh animals yet to be discovered. Nearly 45,000 species of plants and twice as many of animals have been recorded from India. 45 species of plants. .4% land, 8.1% global species diversity, 8.1% global species diversity, 12 mega diversity countries, one of the 12. अच्छी बात है. अभी भी कितना सारा पढ़ना है, मुझे आलसी आ रही है लेकिन पढ़ना पड़ेगा, क्या करूँ. Pattern of biodiversity. Latitude, gradient, species area relationship. The importance of species diversity to the ecosystem. Loss of biodiversity. Habitat loss and fragmentation, overexploitation, alien species invasion, co-extinction, biodiversity conservation. रुको. मैं राइस लेके वापस आ गई। Not uniform throughout the world. The diversity of the plant and animals is not uniform. मुझे पानी भी लेके आना चाहिए था। The diversity of plants and animals is not uniform throughout the world, but shows a rather uneven distribution. इधर जाके खा के आती हूँ, मुझे तुम लोगों के सामने खाने में शर्म आ रही है। कितनी जल्दी खा लिया ना मैंने। मैं अपनी कपड़े के ऊपर बैठ गई।